Welcome to channel Conceptual Biostudy. Dear students, if you like my video, please subscribe it and share it with your friends and others. Thank you all. Today we will discuss about Gymnosperm. Gymnosperm means naked seed. Gymno means naked, that is naked seed bearing plant. Naked seed bearing plant. Okay. According to plant kingdom, we know the plant kingdom is divided into 4 divisions, that is Thallophyta, Bryophyta, Pteridophyta, and Spermatophyta. Spermatophyta, this is the 4th division, is subdivided into Gymnospermy and Angiospermy. Gymnospermy and Angiospermy. Okay, this Gymnospermy divided into 2 subdivisions, that is Cycadophyta and Coniferophyta. Cycadophyta contains one living order, that is Cycadalis, and Coniferophyta contains Coniferalis, Gingualis, and Neetalis. Cycadalis contains the main genus Jamia pygmis. This is the smallest gymnosperm, and Cycas. Okay, Cycas. Coniferalis contain mainly Pinus, Cedrus, Abies, Sequoia, Metasequoia, etc. Gingualis contain living genus 1 Ginkgo biloba, and biloba also contains a extinct genus, that is Gingoitis. This is extinct, Gingoitis. Okay, this is extinct genus. And Neetalis contain Neetum, like Neetum ula, Neetum Nemon, etc. Okay. Now, the term Gymnosperm is coined by Theophrastus. Gymnosperm term is coined by Theophrastus, father of botany. Theophrastus term coined by Theophrastus in his book in Inquiry into Plants. Scientist Goebel called Gymnosperm as phanerogams without ovary. Phanerogams without ovary. Okay. Next, the smell characteristics features. Okay. Gymnosperm are naked seeded plant and they are limited in distribution. They are limited in distribution. Limited in distribution because they grow in cold region. They grow in cold region on mountain slope. Our Indian species grow on Himalayan mountain, slope of Himalayan mountain. As they grow in slope of the mountain, and although the geophytic plants also like to live on the slope of the mountain, so gymnosperms also show that is geophytic characters. Okay, geophytic characters live in cold region. General characteristics. Now we discuss about general characteristics, including the special characters. General characteristics including special characters. General characteristics including special character. Number 1, main plant body, mainly sporophyte, that is twice-in, obviously twice-in, and mainly trees like Pinus, abies, etc., Pinus, cedrus, or woody shrub, like ephedra, or woody climber, like Neetum. Okay, and special type of shrub is found in Welwitschia. That is, here only 2 leaves in the whole year. Okay. Next, some are woody climbers, etc. Number 2, all are heteroecious and dioecious. All are heteroecious. Look here. In our previous class, that is Pteridophyte, mostly homosporous and some are heterosporous, but here all are heterosporous. And dioecious, that is, male and female reproductive structures are found in different plant. Dioecious. Okay. Except Pinus, which is monoecious. Look here, Pinus. These are female cone at the lower base and male cone at the upper side. Except Pinus. Next, they are fast vascular seed plant. They are fast vascular sheet plant. Vascular sheet plant. Sheet plant. You may have heard about CLASS-11 Biostudies. In my previous class I have discussed that they show the heterospory and seed habit, and in this Gymnosperm this trend have been fulfilled here. So they are first vascular seed plant. Next they are vascular bundle. Their vascular bundle is conjoined, unlike Gametophyte where vascular bundle is concentric. Here conjoined, that is they are situated one above the other. Conjoined collateral and open. Open means presence of cambium. So they show secondary growth. Cambium, that is they show secondary growth. They show secondary growth. Okay. Number 5, xylem. Here xylem lac. About xylem, though xylem of Gametophyte and Gymnosperm most cases possess trachete, but here trachete is bordered pit. Trachete with bordered pit. But lack vessels or trachea, except Neetum, Welwitschia and ephedra. Okay. But phloem always lack component cells, unlike angiosperm. Component cells, but they possess the transfusion tissue. Transfusion tissue is present in cycas and pinus. In cycas, look here, this is cycas, this is the pinnate compound leaf, and the pinnae, that is the leaflet, also called leaflet, possess, look here, if it is the cycas leaf and it is the leaflet, this leaflet possess a single midrib only. Look here, this is a single midrib, but there is no vein. Instead of vein, there exists a special tissue which is called transfusion tissue. So presence of transfusion tissue is the characteristic features of most of the cycas or Gymnosperm. Transfusion tissue also present in pinus tissue. Its function is lateral conduction, as there is no vein, so the function is made by transfusion tissue, that is lateral conduction. Okay, lateral conduction. Here some phloem parenchyma cells possess dense cytoplasm which is called albuminous cells or Strasburger cells. That is parenchyma with dense cytoplasm. Okay, cytoplasm. It is a characteristic feature of Gymnosperm. Next, wood. About wood, that is secondary xylem or wood. About their wood may be monoxylic, polyxylic found in cycas, and pycnoxylic or this is monoxylic, pycnoxylic and monoxylic in pinus. Okay, monoxylic means vascular tissue with medullary ray. Secondary xylem absent, so commercially less important. Polyxylic means more than one cambium ring present, or bundle present. Pycnoxylic means look here, pycnoxylic means vascular tissue without or scanty medullary ray with more secondary cambium. So commercial point of view, it is more important, cambium, etc. And monoxylic means only one cambium ring present, okay. Now about their dominant plant body. Next point about dominant plant body, because they are sporophytic and they show diplomatic type of life cycle, so their main plant body is dominant. So dominant plant body. Next point, dominant plant body. Consists of root, stem and leaves. About root mainly taproot. In cycas taproot short-lived and replaced by adventitious root. Okay, adventitious root. Some adventitious root come out negatively geotrophic, and profusely and dichotomously branched. Profusely and dichotomously branched. Leading to distorted coral-like structure, so called coralloid root. These are coralloid root. This is lateral root comes outside and they are branched dichotomously, and they initially infected by bacteria. Initially infected by bacteria, and followed by blue green algae, that is Anabina cycadi, Anabaena cycadi helps in nitrogen fixation, okay. And about the root of pinus, also taproot, root of pinus, mainly taproot, it may persist or converted into adventitious root. Some finer roots, and they are horizontally spreading, mostly horizontally spreading. Some peg-like root comes downward, and the finer roots are two types. Finer roots, two type: normal with root here, and root cap. Some are mycorrhizal root, where scanty root here. So ectotrophic mycorrhiza is found there. Ectotrophic mycorrhiza is found there. That is, fungal attack is found there. The fungus name is Boletus. You know Boletus satanus under Basidiomyces poisonous fungi, but Boletus, some species of Boletus, it's a ectotrophic mycorrhiza. It helps in water absorption, mineral absorption and water absorption. Minerals like nitrogen and phosphorus, and also water absorption. Okay. Now about their stem. Their stem is maybe unbranched or branched. About their stem, may be unbranched as in cycas or branched. Look here, cycas, this is a cycas stem, unbranched, and it is called codex. Because when the stem is unbranched and bears crown of leaves at the tip, they are called codex type of stem. So where branched, there may be, that is dimorphic, in most cases dimorphic, and they consist of dwarf shoot and long shoot. Dwarf shoot, actually they are of limited growth. Dwarf shoot, limited growth, with scale leaves, and arise from the axil of the scale leaf of long shoot. Axil of scale leaves of long shoot. Okay. And about long shoot, they are of unlimited growth. Bears both scale leaves and foliage leaves, that is this is brown color and foliage evergreen foliage leaves. Foliage leaves are photosynthetic but scale leaves non photosynthetic. This is brown in color, this is brown in color, this is green in color, this is green in color. Okay. Now about their about cycas, look here, the cycas, here permanent leaf bases are present in the mature stem. When the young leaves and these leaves, mind it, questions may be from here, the cycas leaves survive few years, that is 2 to 3 years, not many years, 2 to 3 years, and when they dry, their persistent leaf base is present on the stem. Now about their leaves, about the leaves, leaves may be, according to the morphology, may be simple or compound. Simple means when the lamina is either unsegmented or partially segmented, but here compound leaf, the leaf is completely segmented. Look here, in Pinus the leaf is completely segmented. This is the pinnate compound leaf, this is called pinnate compound leaf. Simple leaves present in example simple leaf, example Pinus, Neetum. Look here, in Neetum the leaf is look like the dicot leaf, look like this, just like dicot leaf with dicot leaf. This is opposite decussate leaf with reticulate venation. In Pinus, leaf is very simple, narrow. Look here, this is Pinus leaf, and this is the long shoot, and this is the apex, and this is the dwarf shoot. Look here, this is the dwarf shoot, this is dwarf shoot, and dwarf shoot, look here, in the dwarf shoot there is small scale and it terminates with 3 to 5 foliage narrow leaves, though leaves is needle like, but in sectional view it is semicircular, somewhat semicircular. Okay. Leaves are simple, it is Pinus Neetum. Compound, example cycas, compound leaf, and leaves reticulate, dicot like. Leaves may be scale leaves or foliage leaves, scale leaves or foliage leaves on the basis of function, Scale leaves, protection in function, protection brown in color, protection, it is photosynthetic, that is carry out photosynthesis, Leaves are well adapted to, leaves are well adapted to withstand the extremes of temperature, wind and humidity. Okay, and this show also some geophytic character, especially cycas leaf, Pinus leaf, leaves also show some geophytic character. The geophytic character of leaves like Pinus, cycas, geophytic character of leaves. Geophytic characters of leaf. Number 1, they are reduced in shape. They are reduced in shape. Like needle, like acicular, like needle in case of pinus, needle in case of needle like in pinus, example pinus, or narrow ribbon like in cycas, or narrow ribbon like ribbon in cycas. Number 2, to check respiration, transpiration. Number 1, highly cuticularized, okay. And in case of cycas, epidermis, two-layered epidermis, upper epidermis, parenchymatous, and lower epidermis, sclerenchymatous, okay. Sclerenchymatous. Number 3, presence of sunken stomata, restricted to lower epidermis in cycas, restricted to lower epidermis only in cycas. Sarcinate texis present in pinus. Look here, this is sarcinate texis. There is a fun character, sarcinate texis, to protect the young apical part of the leaf, okay. Sarcinate texis. These are the geophytic characters of leaves. Next, about reproductive structures. Next, we discuss about their reproductive structure. In this context, you should know that the pinus, though most of them perform sexual reproduction, but cycas, bulbil or bud is present on both the male and female mature stem, okay. This bulbil or bud gets detached from the main plant and germinate into new offspring, okay. Now reproductive structures, that is their flowers, as they are gymnosperm, as they are phanerogams without ovary, but flowers is present, though flowers is rudiment. Flowers, first of all, unisexual. Flowers are unisexual. Highly reduced, simple reduced, and that is without perianth. Simple reduced and naked. Naked means without perianth. Naked, that is without perianth. That is no sepals petals. Without perianth, except nitum. Okay. Male flower as unisexual, so presence of male flower and female flower. Male flower represented as microsporophyll or equivalent to stamen, and female flower represented as megasporophyle equivalent to carpel. Now microsporophyll form male cone. Microsporophyll form male cone or pollen cone. Whereas megasporophyll form female cone or sheet cone. Though female cone is absent in cycas. Female cone absent in cycas. Okay, where the female cones are loosely arranged. They are loosely arranged. Look here, loosely arranged around the central axis, around the tip of female plant, Okay, these are the megaspore. Look here, this is leaf-like structure, highly dissected at the apical part and bear 1 to 5 pairs of naked ovule on both sides. Okay, so and about microsporophyll, that is male cone. About male cone in cycas, very large. Everything of cycas is very large. Look here, cycas, male cone greater than equal to 50 centimeter. Occurs singly or in clusters in the apex of main stem, okay, main stem. But in case of pinus, it is 2 to 3 centimeter in length, occurs in cluster. Look here, occurs in clusters at the axil of the scale leaves of the long shoot. That is replaced by dwarf shoot. Actually, from the axil of scale leaves there arise dwarf shoot, but sometimes male cone or female cone may arise instead. Okay. Now about microsporophyll of male cone. Look here, this is the male cone of cycas. It is very large and there it consists of a central axis around which the microsporophyll are arranged in acropetal order. Look here, acropetal order, that means the larger microsporophyll present in the lower part and the smaller one at the upper part. And the individual microsporophyll, if we study the individual microsporophyll, it bears the microsporangia on the abaxial surface. Mind it, microsporangia always present in abaxial surface. In abaxial surface, it is a fun character. Abaxial surface. Okay. In cycas, microsporangia are present in group. In cycas, microsporangia present in group of 4 to 5 leading to formation of sori, leading to form soras. Also, it is the characteristic feature of fern. So, cycas shows so many similarities with fern. This is the microsporangia tetrasporangia. That is, each soras consists of 4 microsporangia. Okay, it's called soras. But in case of pinus, look here, this is a pinus microsporophyll. This pinus microsporophyll is leaf-like structure, consists of a stalk, and the part of the stalk or central axis upper part is called apophysis. The terminal part is slightly bent and it possesses only 2 microsporangia which is oblong shaped and present on the abaxial surface. Look here, in cycas, numerous microsporangia, they are present in the form of soras, but here only 2 microsporangia is present. Okay. And about megasporophyll, look at, there is megasporophyll. In case of cycas, megasporophyll are loosely arranged, but here the megasporophyls are compactly arranged and arise at the base of the axil, at the axil of the long shoot, at the axil of the scale leaf on the long shoot. They are compact structure, woody structure, initially green in structure, but at maturity they become brown, and before just during pollination the megasporophyll becomes open to receive the pollen grain. It is a single megasporophyll. This megasporophyll consists of a woody structure and they also possess bract. If we draw the figure in another way, look like this, look like this, look like this. This is the bract. There is megasporophyll consists of megasporophyll in case of Pinus consists of two scale. This is called ovuliferous scale. This is called ovuliferous scale. This is called ovuliferous scale. Look here, ovuliferous scale, and this is the bract scale. This is the bract scale. Okay, and the ovuliferous, on the ovuliferous scale there is present two wing. Look here, they is present in two wing, and over the wing there is present two invertedly arranged ovule. Look here, the ovule is anatropous. This is advanced type. This is anatropous ovule. But here, in case of, look here, cycas, the ovule is orthotropous. That is, this is orthotropous ovule because micropyle and funicle are in straight line. But here, funicle and the micropyle are in the same line. Look like this, like angiosperm. This is the angiosperm ovule and it also anatropous ovule. Look here, the micropyle and chalaja. This is the micropyle and this is funicle. This is the lie towards the same line. So anatropous ovule is present in Pinus and orthotropous ovule is found in Cycas, and it is primitive, it is advanced type. Okay, these are the megasporophyll and their structure. Okay, now we discuss about in case of Ginkgo, the microsporophyll are arranged in catkin-like inflorescence, that is on a central axis. That is, in case of Ginkgo, this is the long shoot, this is the dwarf shoot. Here bears the scale leaves, and from the dwarf shoot there hangs a pendant central axis where, look like this, the microsporangia are loosely arranged in this way, hanging manner, and each microsporangia consists of two one-lobed anther. This is microsporangia. This is catkin-like inflorescence, and each microsporophyll has a stalk and it bears two microsporangia, and it is a terminal part. This is called the terminal part or hump, and it bears two hanging microsporangia. So these are about microsporangia and megasporangia. In case of, now we study about megasporangium and microsporangium. Okay, this microsporangium may be in all cases the microsporangia is one-lobed anther. Microsporangia is one-lobed structure. Microsporangia one-lobed and consist of multilayered wall and sporogenous tissue. Consist of multilayered wall, and the innermost wall is called tapetum, like that of angiosperm, and this is called microsporogenous tissue. And this microsporogenous tissue, each cell of the microsporogenous tissue converts into microspore mother cell. Microspore mother cell obviously twice n, which on reductional division produce 4 microspores, and this microspores is the first cell of the male gametophyte. About megasporangium or ovule, megasporangium or ovule may be orthotropous or anatropous. First of all, may be orthotropous or anatropous. Orthotropous, that is Cycas, primitive type. orthotropous or anatropous Number 2, in cycas it consists of a massive tissue, this is nucellus, massive tissue that is nucellus, and thick single layer, that is nucellus, and single three-layered integument, that is ovulis, mostly unitegmic. Unlike angiosperm mostly bitegmic, tetrategmic, but here only testa is present, not unitegmic. So unitegmic. These 3-layered means outer layer fleshy, middle layer stony, this is middle stony layer, this is outer fleshy and inner fleshy, this white region, outer stony, middle stony and inner also fleshy, but single layer. Okay. And about in case it is cycas, it is same in pinus also, but in case of gingo is also same, but in cycas and pinus ovule there is vascular supply in the 3-layer of the integument. Look here, this is the vascular supply entered into the 3-layer of the integument, but in gingo no vascular supply in integument, no vascular supply in integument. This is the difference in integument. Okay, this is the difference. And in case of nucellus is, look here, nucellus is fused at the lower part with the integument, but the upper part in most cases forms a big-like structure called nucellar beak. Look like this. This is the micropyle, and this is the nucellar beak, and this is pollen chamber, and this is nucellar beak, this is pollen chamber, and nucellar beak possesses pollen chamber, and during pollination this is the archegonia, this is archegonia. Okay. This in most of the cases nucellar beak present and pollen chamber present in cycas and gingo, but absent in pinus and nita, Okay. Now megasporogenesis. Now about megasporogenesis, that is development of megaspores. Megasporogenesis. Megasporogenesis. Now, the in case of cycas, in cycas, only one, in cycas only one or two in gingo, one or two in gingo, or several in neetum, or several neetum, that is within the nucellus only one or two or several megaspores mother cells can differentiate. Okay, this megaspores mother cell undergoes reductional division and they form four megaspores. Of the four megaspores, only three gets degenerated, three degenerates, one remains survival, one remain functional, that is functional megaspore. Example, functional megaspore. Example, cycas pinus. Gingo. But in neetum, out of four megaspores, all megaspores acts as functional megaspore. Okay, functional megaspore. These functional megaspores form female gametophyte. And this type of development of female gametophyte or embryo sac is called tetrasporic, as four functional megaspores form gametophyte, so it is tetrasporic, found in some gymnosperm, angiosperm also, tetrasporic. But here the development of female gametophyte is called monosporic. So in case of gymnosperm, mainly development of female gametophyte is monosporic. In some cases, the advanced genus, that is Neetum, tetrasporic, like that of some angiosperm. Okay. Now about male gametophyte, development of male gametophyte. Development of male gametophyte. First, microspore is the first cell. Microspore is first cell of male gametophyte. Number second point, it develops initially within microsporangium. Okay, within microsporangium. First, each microspore is with two layer, outer exine and inner intine, thin intine. Exine is thick and intine is very thin. Okay, and within the microsporangium, each microspore first cuts off by mitotic division, cuts off two cell, that is one permanent prothelial cell in cycas, permanent prothelial cell, small, and large antheridial cell, antheridial. This is found in cycas, gingo and nitum. Cycas, gingo and nitum. Cycas, gingo and nitum. Okay, or it may divide two prothelial cells, or it may divide initially two prothelial cells. One is permanent and one is degenerative. One is permanent and the rest one is antheridial cell, as in pinus. Okay, and after this, that is 2 cell stage or 3 cell stage, or 4 cell stage. These antheridial cells further divide into two. Look here, that is one permanent prothelial cells and antheridial cells, or two prothelial cells, one of which may persist, and the antheridial cells in pinus. Okay, and in nitum, the dehiscence, antheridial cells by further divisions, mitotic division, forms a smaller generative cell and larger tube cell. Okay, and dehiscence in 3 cells, cycas, gingo. Dehiscence takes place in 3 cell stage or 4 cell stage. 3 celled or 4 celled. 3 celled cycas, 4 celled pinus, gingo, etc. Pinus, gingo, it's cycas, nitum, 3 cell stage. Okay, and after dehiscence, the further division takes place in the ovule. Okay, next we discuss about the further development. Further development on ovule, that is further development of pollen grain, that is male gametophyte. Okay, here the tube cell, larger tube cell elongates when after pollination. Look here, when sits on the apex of the ovule, there is a drop of mucilage which is called pollen drop, where pollen drop, and this pollen drop, when pollen drop, it is present in pollen chamber and it is caught by the pollen drop, and when pollen drop becomes dry, it tube cell elongates and form branch tube or unbranched tube. Tube cell elongate. But here point to be noted that after pollination, the further growth may not may not continue in case of pinus, because after pollination the further development or fertilization takes place about 13 months later, that is about 1 year later. Okay, but in case of other genera immediately or few days later the fertilization takes place. So here the tube cell elongates and form branched or unbranched pollen tube. Branched example cycas and gingo, or unbranched pollen tube. Mind you that in Gymnosperm branched pollen tube is the characteristic feature. Unbranched pollen tube example pinus and neetum. Okay, pinus and neetum. These pollen tubes at the same time germinal cells divides mitotically from stalk cell and body cell. But in case of neetum, the antherial cells directly, that is germinative cell directly form two non-motile gamete in case of neetum. But in other genera, germinal cell for the first time divides into stalk cell and body cell. Stalk cell, it is degenerative, it is functionless, and body cell further divides into motile or non-motile male gamete. Motile gamete in cycas and gingo. And non-motile in pinus neetum. Okay, in this way full development of gametophyte takes place over ovule. Okay, next we study about the pollination. Development of female gametophyte. Development of female gametophyte. Look here, megaspore, that is functional megaspore is the first cell. Is the first cell of female gametophyte. Initially the nucleus of female gametophyte, look here, this is the female gametophyte nucleus. By repeated free nuclear division, it produces numerous nuclei. Nucleus by free nuclear division, numerous nuclei. Suddenly there appears a central vacuole. Look here, there appears a central vacuole. Look here. And next, movement of nucleus and cytoplasm towards periphery. Cytoplasm with nuclei moves in periphery. Okay, periphery. Then next, wall formation laid down between the nuclei. Wall formation laid down, wall formation is laid down between two nuclei centripetally, that is centripetally, that is from periphery to center. Look here, this is in this way, and ultimately the entire cavity of the megaspore is filled up with a solid mass of tissue. Next, the entire cavity of megaspore filled up with solid mass of tissue called endosperm. So endosperm is obviously haploid because megaspore is haploid, and this endosperm tissue is formed by mitotic division, free nuclear division, then cell wall formation. So endosperm is haploid and formed before fertilization. It is a characteristic feature, unique features of gymnosperm, formed before fertilization. Okay, fertilization. Some endospermal cells towards the micropylar region, forms archegonia. Some endosperm cells form archegonia at micropylar end. Look here, this is two micropylar end. It may be 2 to 8 in cycas, 2 to 8 in cycas, 2 or rarely 2 to 3 in pinus, 2 to 3 in pinus, and no archegonia in 2 or 3 in gingo, and no archegonia in neetum. Very advanced archegonia in neetum. Okay, neetum is formed at the tip of the micropylar region from the endosperm cells. Okay, and now about pollination. Each archegonia, where archegonia is present, each archegonia consists of 2 or 8 celled neck, 8 cell neck and 1 ventral canal cell and 1 egg. Okay, each archegonia consists of 2 or 8 cell neck, 2 cell neck cycas, 8 cell neck in pinus. Okay, 8 cell neck pinus. No neck canal cells is present. Neck cells, but no neck canal cell. No neck canal cell is found here. That is, the archegonia is very much reduced, unlike Pteridophyta and Brachyphyta. Okay, and next about pollination. Next about pollination. Pollination, it happens in 3 to 4 cell stage. It occurs in 3 to 4 cell stage by wind. Pollination by 3 or 4 celled male gametophyte male gametophyte by wind, most cases wind, that is anemophily, or by insect in Neetum and Ephedra, Neetum and Ephedra. Okay, and in Pinus, a special type of pollination is found in Pinus, that is shower of sulfur is found. That means shower of sulfur. Look here, this is the Pinus pollen grain, and exine and intine. Exine is prolonged and extend to form the wing. This is the wing, acellular layer wing, and on the base of the yellow in color, when numerous yellow colored winged pollen grain are transmitted through air from one hilly area to other hilly area, looks like shower of sulfur or dust of sulfur. Okay, dust of sulfur. Next, about at the time of pollination, pollen drop oods out, already discussed. At the time of pollination, pollen drop oods out from the microphylla region, which helps in catching the pollen grain, that is gametophyte, 3-celled or 4-celled gametophyte, and helps to proliferate or produce pollen tube. Fertilization. Next about fertilization. Fertilization, it is two type, by judeosiphonogamy and siphonogamy. That means when in cycas, look here, the cycas, pollen tube, that is branch pollen tube, and within the pollen tube there runs two multiflagellate top-shaped motile gamete through pollen tube. So it is called judeosiphonogamy. That is present in cycas and gingo. Example, cycas and gingo. But when non-motile gamete, only non-motile gamete passes into the pollen tube, non-motile gamete, then it is called only siphonogamy. Example, Pinus, Ephedra, Neetum, etc. Etc. Okay, and during fertilization, pollen tube grows downward. Look at pollen tube introduces into the pollen chamber, where pollen chamber is found, and ultimately the neck ventral canal cells dissolves and introduced, they get burst, the tip of the pollen tube get burst and discharge their contents within the pollen tube or archegonia, or directly at the tip of the micropyle, as in case of Neetum, because there is no archegonia. Ultimately one of the male gamete, look here, one of the male gamete fuses with the one of the male gamete fuses with the male gamete, obviously N, fuses with the egg, haploid in number, leading to form zygote. Okay, and it is the first cell of the diploid stage or sporophytic generation. Then the development of zygote, that is embryo formation. Development of zygote. Look here, development of zygote is not done by vertical or transverse wall formation, not done by, unlike angiosperm, not done by transverse or vertical wall formation, transverse or vertical wall formation. Vertical wall formation. Instead, it is done formation. Instead, it is done through prenuclear division. Through prenuclear division, unlike angiosperm. Prenuclear division. Okay, nuclear division. Next, embryo formation. In this way, zygote converts into embryo. Now about embryo. In major cases, embryo is polyembryony. Formation is called phenomenon of producing many embryo, that is polyembryony. Here, but no polyembryony in cycas and gingo. First of all, no polyembryony in cycas and gingo. But in pinus and some species of Neetum polyembryony found. Polyembryony, that is more than one embryo, polyembryony found. Polyembryony is of two types, that is simple or potential polyembryony, and true polyembryony or cleavage polyembryony. In simple polyembryony, embryo develops from archegonia. Embryo develops from archegonia. So more than one archegonia, more than one embryo formed. Archegonia. But here more embryo formed by mitotic division of zygote. That is zygote, one zygote split, two zygote, and this form embryo. And this further split into another segment. Each segment form embryo. Okay, embryo. So polyembryony is found in pinus, etc. Okay. Now embryo, polyembryony, mature embryo. Now about mature embryo. Parts of mature embryo. Mature embryo consists of radical, plumule, and two cotyledon as in cycas, and also neetum, but they are radical with root, where root with root cap, extra character, root cap. Okay, or it may be radical hypocotyle, 3 to many cotyledon, Look here, here 2 cotyledon, here 3 to many cotyledon, as in pinus, many cotyledons and plumule as in pinus, okay. Or it may be radical plumule and 2 to 3 cotyledons as in gingo, okay. These are the type of embryo. Now about seed structure. Seed may be not winged, in most cases not winged without aerial. Aerial is the sometimes as in found in lychee, maize, where the nucellar part comes outside of the seed coat and forms a fleshy structure which is called aerial. But here in most of the cases without aerial and colored fleshy seed coat, fleshy seed coat as in cycas, and due to presence of colored fleshy seed coat, it is attracted by birds and helps them in seed dispersal. It may be consist of not winged and large with seed coat, not winged and large seed coat which consists of 3 part: outer fleshy, middle stony, that is hard layer, stony, and inner fleshy as in peppery, inner peppery nitum, as in nitum. But seed may be winged. Look here, this is the winged seed. As ovule develops over wing, and ovule when becomes converted into seed, still now seed bears wing, that is winged seed, in case of pinus, winged seed, and without aerial as in pinus, okay. Now about their life cycle. Life cycle is diplontic, that is due to short-lived gametophytic stage, gametophytic generation. Here represented in diagram, that is male sporophyte. One plant is as there is dioecious, so male sporophyte, obviously twice n. It produces male or microsporophyll, diploid. It produces microsporangia, twice n. It produces microspore mother cell, microspore mother cell twice n. It undergoes reductional division, produces microspore, that is the first cell of the male gametophyte, microspore. Okay, in this way, this is another plant, female sporophyte. Female sporophyte, obviously twice n. It produces megasporophyll, twice n. It produces megasporangia, twice n. It produces megaspore mother cell, twice n. Undergoes meiotic division, it produces megaspore, functional megaspore. It produces male gametophyte. On germination, male gametophyte. On germination, it produces female gametophyte. It consists of archegonia or egg in case of neetum, and endosperm. Both are haploid. It produces male gamete, motile or non-motile, motile or non-motile, then male gamete, and female gamete by fertilization through siphonogamy or judeosiphonogamy. Fertilization, it produces zygote. Zygote by mitotic division and development, it produces embryo. On germination, it produces male gametophyte or female gametophyte. Female sporophyte or male sporophyte. So it accomplishes through two different stages, that is this stage it is sporophytic generation, it is gametophytic generation. Okay, gametophytic generation. Now discuss about economic importance, that is last topic, economic importance of gymnosperm. First point, economic importance. Number one, cycas leaves are used in making mats. Young cycas stem, young shoot and seed are cooked as vegetables. Okay. Number two, a starchy food obtained from Cycas revoluta revoluta, obtained from stem of Cycas revoluta, as sagu, in the form of sagu, so it is called sagu palm. So Cycas is called sagu palm. Okay. Number 3, various species of Cycas revoluta. Various species of Cycas, that is Cycas revoluta. This revoluta also used as ornamental plant in the garden, and another Oracaria under Coniferaceae, Oracaria heterophylla, which is called star pine or Norfolk Island pine. Heterophylla also as ornamental plant. Okay, ornamental plant. Number 5, seeds of Pinus gerardiana, that is Chilgoja pine. Seeds of Pinus gerardiana, which is called Chilgoja pine. Pinus gerardiana, Chilgoja pine, Chilgoja are used after roasting. Okay. Next, about some species of Pinus, that is Pinus roxburghi, what is called chirpine, roxburghi. Okay, and Pinus walliciana, and cedar plant, that is Cedrus deodara, plant, not cedars, deodar plant, that is Cedrus deodara, are important timber yielding plant. Here the timber is soft and used as making light furnitures, as electric pole, Pinus pole, especially Pinus wood, and matchsticks, railway sleepers, etc. Okay. Next point about resin. 6, 7. Resin, it is extracted from Pinus roxburghi and insularis, and Pinus insularis. Resin is the semi-fluid, extracted commercially from resin duct or resin canal, resin canal, and they are used as sealing material, as inflammation, to check inflammation, sealing joint, and here just a minute before I have told you that before fertilization the megasporophyll opens, and after fertilization they becomes sealed by this resin. Some resins, when these resins after distillation, form distillation, it converts into turpentine, which is used as the base of solvent, used as solvent for paint, polishes and wax. Okay, next about amber. It is a fossilized resin. Amber, it is fossilized resin from Pinus succinifera, extinct member succinifera. It is used in jewelry. It is used in jewelry, and it is important. It has important commercial value. Next about Taxol. It is an anti-cancerous Taxol, anti-cancerous agent extracted from Taxus, that is commonly called yew plant or yew tree. Next, cedar oil from Juniperus virginiana, that is extracted from its bark. It is used as cleansing fluid in biological laboratory, and to increase resolving power of microscope, and it is also used to remove nail polish remover, as nail polish remover. Its wood is used as pencil manufacturing. Wood is used in pencil manufacturing. Okay, so this is all about gymnosperm. Thank you all.